So our next speaker is Professor Catherine Cole. She's the inaugural Professor of Paediatric Haematology and Oncology at the University of Western Australia. She's got extensive research background managing children with leukemia and cancer in randomised controlled trials of the Children's Oncology Group, which is an international cooperative group which prides itself on remaining at arm's length from pharma.
Thank you very much. These are my affiliations, and it is the largest cooperative group studying children's cancer that I've been working with in the COG. These are my conflicts of interest, and this is what I try and teach medical students, and I should disclose that I sit on the PBAC, and as we've recently been discussing and will continue to discuss a lot of very expensive new cancer drugs, you'll see that it's flavoured my talk. I'm going to start with these words from 40 years ago now, one of the doyenne of paediatric oncology, Audrey Evans, and she pointed out that even at that stage, paediatricians had really led the way in treating people with cancer because we had defined the two essential ingredients, one being collaboration with all our fellow disciplines, and the other recognising that cancer in children was very sensitive to the drugs that we had at that time. I think that I'm going to show you how important the first of these is, collaboration, but I don't want to belittle how much we look forward to even better therapies, and around about the same time, Dan D'Angio was already very prescient in saying we await for these targeted therapies so that we'll have fewer long-term effects of therapy. The disease that I've spent most of my career treating is acute lymphoblastic leukemia, the most common cancer of childhood, and as you'll see from this graph, apart from after we discovered we needed to address the central nervous system in the early 70s, there's been no miracle in the improvement in treatment, but rather a slow stepwise improvement with each series of trials building one on the other, and that's an important point. The story began way back before the 1950s, and the details of it are in this book that I hope you've all read. It's a magnificent story, and it began with the antimetabolite amenopterin. In the 1960s, it was realised that we could treat children's leukemia in much the same way that we'd treated tuberculosis, with a combination of therapies so as to avoid resistance and to have non-cross-reactive side effects. From the very beginning, we were already talking about prognostic factors and putting our patients into particular groups according to their individualised characteristics. So I think we've always practiced personalised medicine. It's just that the bits of science we've used to do that have changed and been added to over time. In the 60s, there was already a bit of happy rivalry across both sides of the Atlantic, arriving at the same sort of treatments, again with these multiple treatments of non-cross-reactive therapies to cure children with leukemia. Along the way, we've always been hand in hand with our basic scientist laboratories, with specimens being stored. We've had tumour banking from the very beginning. In the 70s, we learned about cytogenetics and how that could affect our prognostic groups of patients. We were adding new drugs, and as you can see, we couldn't have done any of this without the role that statisticians have played. By the 1980s, it was truly more personalised medicine. Here, the median survival was over 10 years, and we had more risk groups that we could put patients into and adapt the treatment accordingly. We were also looking at the way each individual metabolised the drugs so that we could adjust treatment for each patient. By the 90s, because our patients are with us and we continue to follow them as time goes on, we were looking at the long-term effects of therapy and feeding that back into the way we treated patients. We were able to tweak a few of our drugs a little more and get even better results. But you can see that all of the drugs that we were using at that stage had been around since the 1960s, and that the improvements had actually come from better risk-directed therapy and, of course, supportive care and being able to keep our patients alive. All of this was done through randomised trials. Into the 2000s, and now we were able to look far more closely than just down the microscope as to when a patient went into remission. We can now look to the level of 1 in 10,000 cells and decide whether a patient has gone into an early remission and therefore requires less intense therapy than someone who still has minimal residual disease. So the improvement in outcome in these 5 decades was cemented in the paradigm of treatment for all children in cooperative group randomised controlled trials in paediatric hospitals. It puts the patient's need to be cured first and recognises that the best chance to be cured is the first time. That's the bleeding obvious, you're going to say to me, but actually it's really important, and it explains the emphasis in paediatric oncology of putting patients on phase 3 clinical trials and spending much more time on that than on phase 1 and 2 drug trials. So what I've described to you is this: the patient comes in, they're diagnosed, they go on a trial, and hopefully they become disease-free. We do not use the term standard care, something the Americans call standard of care. I don't know what that of is there for. I admit that there are patients who will turn up with metastatic disease and patients who do have relapsed disease, and that some of these will go on and die. And many of these patients are, I think, very altruistic in that they do go on drug trials, phase 1 and 2 trials, which are essential for getting out those new drugs down the pipeline into upfront therapy. However, I think that we still need to concentrate where the money is, and that is in curing patients on phase 3 trials. I've made the point of saying some of the numbers here about that you need in order to do these studies. Very few patients are needed for the data for phase 1 and again more but fewer than in phase 3 trials for phase 2 trials. And that may well lead you to think, well, they must be very expensive, thousands of patients on those phase 3 trials. And perhaps that explains why the drug companies do this. They market drugs from phase 2 for patients with relapsed metastatic and high-risk disease. They come and see us at the PBAC and they talk about the very high unmet need of these poor dying patients. And I don't want to belittle the fact that we need to care for patients with end-stage disease, but I put it to you that the area of unmet need is in fact in doing many more of these studies. Therefore, I'd like to say that we no longer talk about investigator-initiated clinical trials because that's a very cumbersome mouthful, but that we take back the term clinical trials and insist on calling these trials drug trials. These are clinical trials. I'm not the first person to say this. A few years ago, 25 years after the Journal of Clinical Oncology was begun, the editors looked back and looked at what had happened in the field and whether things had been achieved that they'd hoped for when they launched the journal. And they were a little sad, and one of the things they said was oncologists should reduce their participation in phase 2 trials and be given more academic and other credit for supporting phase 3 trials with the potential to change practice. In Australia, 2/3 of trials in cancer are funded by industry. They're nearly all in metastatic disease where therapeutic gains can be more promptly translated into commercial success. Now, in the paediatric centres, and certainly the 2 most successful recruiting centres in Australia are ours in Perth and the centre in Brisbane. And at any one time, we have trials open for about 2/3 of our patients who walk in the door. Over 95% of them patients are then enrolled on those studies. Well, that must be very expensive, but I'll show you it's not. The National Cancer Institute spends $25 million running the Children's Oncology Group. So that's the first thing. The American taxpayer actually pays for all this anyway, not us. But that comes down to about $70 per year of life saved for every child who's on those trials. Now, the other third of patients who are not enrolled on trials in our units also benefit from the fact that they're being treated in a unit that runs clinical trials. So that's $40 per year of life saved. So the total cost of running these cooperative group trials is about 1% to 2% of the total cost of cancer care. So you're going to say, well, yes, Kathy, but cancer care is really expensive. Well, it's not. Here's the cost of treating and curing childhood cancer compared with other things that we think are worth spending money on. I often show this data to middle-aged male hospital executives. They're particularly keen on thrombolytic therapy for acute infarcts. So I've shown you then that clinical trials are cheap. There's no reason why we shouldn't be doing them. This is the saddest slide that I show. This is different from the Kaplan-Meier plots I was showing you before. This is a population-based study over time, and what it shows that indeed the different biologic groups of acute lymphoblastic leukemia have different outcomes. But what you can see is that the preschool children, the primary school-aged children, the young teenagers, and even that tricky group, the infants, have improved over time. The slope of those 4 lines is the same, and that's because those patients have all been enrolled on clinical trials of the cooperative groups. What's going on here? These are our older teenagers and our young adults, and the problem here is that they walked into the wrong hospital door. They did not enter a paediatric hospital, and they got standard of care. As I look around the audience, I see that you, like me, probably have children in this age group, so I find this not only sad but terrifying. We can do amazing things when we work together in a cooperative group. This disease, Philadelphia chromosome positive ALL, is really rare in children. It's much more common in adults. We had to get together with our colleagues on both sides of the Atlantic in a nice town in Italy to look at this disease, and in the pre-imatinib era, it was very clear from pooling our data that these patients would only be cured by transplantation. So imatinib came along, we were really excited, but look at all the patients in the adult sector. Who do you think did the trial? We did. And this was probably an early adaptive trial, to use the trendy language of today. There were 5 different cohorts to get the dose right of this new drug, and from very early days we could see this was a game changer, and the toxicity of transplantation was no longer required to cure this group of patients. So what's the future? Well, our very good risk patients now do so well that the statisticians tell us it's going to be incredibly hard to improve things, but we'll keep trying. But of course our efforts need to go with those patients who still relapse. And here we do have cooperation from industry, because, you know what, they've realised that paediatric oncologists are really good at running clinical trials. It would be remiss of me not to mention most of the world that's not included in these large cooperative groups in wealthy countries. But here too, I think paediatricians have shown the way in being able to put a statement out that the outcome for acute lymphoblastic leukemia in children is now so good, this treatment must be available to all the world's children. And indeed, we have the 10 drugs required now on the essential medicines list of the World Health. I've gone over time, so I'm going to leave you to read these final words of Audrey Evans, and even though I fear sounding like President-elect Trump, I think, ACTA, we need to take back clinical trials.
Question over here from William. Rhiannon? You go.
William Tanamuri from NHMRC Clinical Trial Centre in Sydney. Thank you very much. That's really inspiring.
Thank you.
And it's an example that needs to be really used again and again. It does illustrate, I think, something that's become more generally known through prospective studies and phase 3 trials, which is that you're actually better off being in any phase 3 trial, even if you're in the control group, than not being in a phase 3 trial. And I think one of your sad slides shows that. Mm-hmm. And I just wondered, and I'm particularly aware of the fact that we have members from the Bellbury Ethics Committee, for which we have the highest respect, whether the time has come for ethics committees to actually take their responsibility of conveying the benefits of research as well as the risks of research by translating this kind of information into non-coercive but clear messages that have to be relayed to prospective patients in trials on hospital websites and research ethics forums.
I agree with you entirely. I like to say to my ethics committee, why do I get the feeling that you're trying to stop patients going on trials rather than roaming the wards of the hospital finding out why there are so many patients not being treated on trials? They don't like it when I say that. But I think Steve got it right this morning when he said that we should stop considering all sorts of trials the same way, and these ones should have very streamlined ethics and governance approval.
Okay, thank you very much.